amino acids, okay, the different R groups of amino acids, which are like the functional groups, and peptide bonds. The guided question I want you to keep in mind today is how do amino acids help determine protein structure and function? Okay, so as just a repeat, amino acids are the building blocks, okay? They are the monomers. Now they have different properties. They could be non-polar, polar, negative, or positive. So if they're negative, they are acidic, and if they're positive, they are basic. Okay, the sequence matters. So the sequence in which these proteins arrange themselves matters because that will determine the secondary structure. And when the secondary structure is folded, it will become the tertiary structure. And if there are a bunch of tertiary structures coming together, it forms a quaternary structure. This we will do a lot in the next lesson and the different protein sequence levels. And then very important today is that shape determines function. Okay, so enzymes speed up chemical reactions, and they can speed up these chemical reactions because of their specific shape. If the shape changes, they will not be able to perform their function. Okay, it might be slowed down or stopped entirely. Proteins are also transport proteins. Okay, so to transport things across the cell membrane. If that structure changes, it might interchange how these molecules interact with this transport protein. And then finally, they're also structural proteins to provide support and shape. Okay. So if we start with proteins in a big view, so amino acids connect through a dehydration synthesis reaction. Okay, so we have an OH group and an H group that will be removed to form water. And once the water is removed, it forms a peptide bond. And these peptide bonds are covalent bonds. Okay. So the general pattern here is an amino acid plus amino acid plus an amino acid makes a polypeptide chain, and this is the polymer of proteins. If we look at what is an amino acid, we can see that each amino acid has a general structure. Okay, so this part of the amino acid is the same for all amino acids. It has a central carbon, okay? It has a hydrogen molecule that's attached to the central carbon. And then it also has an amine group, which is known as the N-terminus. So this is the N-terminus. And this is the C-terminus because it has a carboxyl group. So this is a carboxyl group, okay? This is the hydroxyl group that would, in a dehydration synthesis, be removed. And then finally, in all amino acids, this is the R group, and this is different. So each amino acid has the same backbone, and they have different R groups. Now, the hydrophobic or non-polar R groups, they do not interact well with water, okay? And they often move away from water. So if I have a protein that has these hydrophobic groups, okay, those proteins will kind of fold in to the center of the protein to be further away from water, okay, because they don't like interacting with water. So they would generally be, if we look at the 3D structure of a protein, they would generally be found on the inside, okay. Hydrophilic groups or polar groups, these groups would interact very, very well with water because they are polar molecules. And usually we find these on the surfaces of proteins. Okay, ionic groups have a charge. Now, this charge can either be positive or negative, and they can interact with other charged regions. So if there is a negatively charged molecule, it would interact with that positive part. And if there is a positively charged one, it will interact with the negative part. It will also interact with water and also other polar regions. So for a visual check, check the following scenario. Look at the folded protein in water. Now, which R groups are likely to be buried inside the protein? The ones that are non-polar or hydrophobic, okay? So all my non-polar molecules or R groups that are non-polar would tend to be on the inside. The ones that tend to be on the surface, these could be my polar ones, my polar groups, okay? Because they really interact with water well. They would form hydrogen bonds with water, okay? And then finally, why would non-polar groups be pushed away from water? Because it doesn't interact well with water. It doesn't form any connection with water. So it's like lipids and water. It tends to form a separate layer. And because it forms a separate layer, they kind of like fold away into the center of the protein. And like, how can this affect the protein structure? Okay, so it affects the protein structure because the hydrophobic molecules tend to be on the inside and hydrophilic molecules tend to be on the outside. The essential knowledge for today, okay, proteins are built from amino acid monomers. Okay, so these are my different R groups. So these are all different amino acids. And these are my monomers, which are amino acids. And then amino acids are connected by a covalent peptide bond. So when these two amino acids join, they form a covalent bond and it releases water. Okay, because this is a dehydration synthesis reaction. And each amino acid has a central carbon, a hydrogen atom, amine group, carboxyl group, and this variable R group. And then finally, these R groups can either be hydrophobic, non-polar. They can be hydrophilic, polar, or ionic. And ionic just means it has a charge, a positive or negative charge. If you enjoyed that video, please don't forget to like and subscribe, and can join us on the Skool.com community page where you'll find AP Bio blueprints. And if you take a look inside, you will see that Unit 1 is completely free. See you all there.